Alright, Unit 7, Carbohydrates. This unit is a little bit easier, I think, a little bit more easier to grasp. So, definitely know what carbohydrates do. They're most abundant organic molecules in nature. Always be able to classify them by mono, oligo, or polysaccharides. Two main types. So the carbonyl carbon, or the functional carbon is the other name it's referred to. For the aldoses is the first carbon, and ketoses it's the second carbon. So in nature we have D-isomers. That doesn't come up too much. We'll kind of see examples of this a little bit later on. He'll almost guaranteed to ask about dihydroxyacetone. It's the only monosaccharide that is not chiral. So think of it kind of like glycine. No stereoisomers. So this usually comes up on quiz questions. I would just know the difference in how to identify these. So constitutional isomers, they're totally different. They have the same exact formula, but the structure is totally different, is what I mean to say. Epimers are just one carbon difference. So glucose and mannose are epimers. Lactose is another epimer in there, but we'll look at that in a minute. Anomers just means that the alpha or beta is going to be different. Enantiomers is D and L are different. And diastereomers, they are look the same, but they're not mirror images. Isomers, but not mirror images. A little rusty on that. So this, I wouldn't call this top priority stuff, but good to know, especially for a quiz. So here's a couple common ones. First chart to definitely memorize, important monosaccharides. Just remember these are functional carbon 1 or carbonyl carbon 1, and these are C2. So the most common thing you'll see is the whole question will say something like ketopentose. And that's the question. So what's the ketopentose? It needs to be first from the ketone group, and secondly 5 carbons. So one of these would be the answers. Don't mix up ribulose with ribose. Let's see. Trying to think if I had any good stuff to memorize here. Dr. XF's is how I would always write out this. And GERD, followed by the epimers, GGM. We'll talk about why these are the epimers there. So, know this whole thing and know that he might say aldotriose. So what's an aldotriose? Aldehyde of 3, glyceraldehyde. So these are the epimers that you have to worry about. They're both based off glucose. Mannose, the one with 2 N's, is the C2 epimer. Galactose is the C4 epimer, just meaning at that position they're switched, mirror imaged. I don't see much questions on the hemiacetal and hemiketal, and the cyclic forms aren't asked about much either. I would know that Pyran is 6 rings and Furan is a 5-point ring. This one is pretty, I see, asked in a couple different ways. So in a fructose solution, purinose. So predominates in a fructose free in a solution. So when you have pure fructose, you have purinose. That's how I remembered it. Furanose is in derivatives, so that's the more common form, but if you have pure fructose, purinose. So make sure to know that gluconate is oxidation at carbon 1, glucuronate oxidation at carbon 6. This is some more lab stuff. Just know that they react with copper. This is some more like, I guess, higher level stuff, that some of these, that you can add different groups onto the sugars, just like you can add different groups onto proteins. I might know that these are bigger than your traditional monosaccharides because they have something added onto them. If you were given all these on a quiz, this one would be the biggest because it not only has this added on, but it has this R group. This R group here is this. Yeah. This R group is this here. Again, quiz level. Sugar phosphates, when you're asked what's larger, phosphate is relatively huge, so especially with if you're given a bunch of monosaccharides and one of them has a phosphate attached, that's your answer. That's a very, very good tip that helps you quite a bit, so remember that. Don't need to worry about that too much. So for your disaccharides, just be able to name the bonds for all of them. We'll see a better chart of it coming up here, so don't worry about that too much. For now, we'll find it. Let's see, starch is made of two main things, 20% amylose and amylopectin. And for these, you're thinking, are they linear or are they branched? So 80% of starch is amylopectin, so 80% of it has these branches in it. So we'll take a look at how he asks those questions here. Alright, good stuff on starch. So we know amylose is 20% and it's linear. Amylopectin is 80%. Every 4% of them branch. So if you were to write this out for amylopectin, it is only 4% branched. And it's 96% linear. Branched needs to be in your brain synonymous with alpha-1,6. Linear needs to be synonymous with alpha-1,4. So that's, if you're asked about amylopectin, that's what it is. Amylose is 100% alpha-1,4, meaning 100% linear. If you were asked about the composition of starch, if you were to do the math out, because starch is mostly amylopectin, but it also has some extra linear, starch is going to be 97% linear, alpha-1,4. Which makes it 3% branched, because it's your amylopectin here, which is a little diluted with more this linear amylose. So these are going to be your four questions, or your four options most likely. You have to know if he's asking about starch or amylopectin, and if he's asking about linear or branched, or what the linkages are. So know that really well. Glycogen, 10% branched, 10% alpha-1,6, 90% alpha-1,4. Know about where it's found and what it's used for. It's just a bunch of glucoses in a row. Cellulose, most abundant compound in the world. That comes up sometimes. These are beta-1,4 linkages. We'll see a chart that summarizes all these for us too. So glycoproteins, here's another little subsection. We're going to talk about all these for just a minute. And on my study guide sheet that I have for you, I take out the main points, such as this, for what you have to know. N-linked versus O-linked. The EPO is an example of a glycoprotein. Proteoglycans are your GAGs, mostly protein by weight. These are some examples of them. Maybe be familiar. I think heparin comes up more than the other ones. These ones with sulfate are pretty heavy as well in a question like that. Heparin has these 3 sulfate groups, so it's a heavy guy. So cartilage is another proteoglycan. Mucins and mucus. Again, refer to the study guide for the most commonly asked stuff. Chitin, I love these slides. So they don't tell very much. Chitin is like in the wings of these guys, so that's another form for another use of carbohydrate. Let's see, so this is your good chart to know about polysaccharides. It summarizes everything you need to know about them. Definitely know the repeating unit, telling the difference between all of the linkages. They're all homopolysaccharides except for this guy down here. This is glucosamine. Make sure you know that. These are all glucose here, but this last one has chitin and GAGs have glucosamine in them. The role, I would know that as well. Alright, this is guaranteed a couple questions at least, and you kind of just have to know everything on there. So know what substrate is often going to be referred to as targets. So let's walk through this. You put a piece of bread in your mouth and there's starch. So the amylase in your mouth, and you have some backup in your pancreas, the amylase is going to break down the starch, so that's its target, and into dextrins. Dextrins are these 3 guys here. So these 3 dextrins are going to become the targets for these. So these 3 enzymes, maltase, glucoamylase, isomaltase, these all are targeting dextrins, because these are the dextrins here, and these are their targets. Oftentimes everyone makes the mistake, they say, which one targets dextrins? And they say starch. Starch doesn't target dextrins. Starch produces dextrins. Invertase and sucrase are used interchangeably, so know them both. Remember, these are all small intestine except for the first one. And lactase targets what's usually might be called like an animal carbohydrate or something like that, lactose. And it's the only beta-1,4. First 3 on the list are alpha-1,4. You have this weird alpha-1,6, and then sucrose is alpha-1,2 link. So know everything on there, but be able to understand it too. Very common questions that come from here. Make sure to note if what's happening. Are they being absorbed, released, both? No, glucose and galactose are always bonded together here. Remember, glucose and galactose make up lactose. So a question might be like, which transporter releases the product of breaking down lactose? So which transporter, when you break lactose down to glucose and galactose, releases the product, GLUT2? That's how he's going to ask this sort of question. And this one has 3 molecules involved, sodium, potassium, and ATP. And we talked about this at the end of Unit 4, that sodium amino acid symporter. Remember, GLUT5 does fructose, and it does both. This is a tough one. I would know this pretty well. So we got in cells, out of cells. The most important thing you can probably do is know the second highest and lowest for both of them. So the lowest, if it's ever an option, is going to be your proton. It's such a small amount. Your second lowest for in the cells is calcium. If you're going to go the highest, it's going to be potassium in the cells or sodium out of the cells. And then second highest, it's kind of too hard to tell in the intracellular, but you maybe know chloride is the second highest extracellular. So work with that chart, try to find out some way to know those really well. It usually pops up for one question. Lactose intolerance, very prevalent, so keep that in mind. And that's preview for Biochem 2.